Hi everyone. I am privileged to be here today, and I'd like to thank the organizers for giving me this opportunity to deliver my journey in science to you. I know not many of you are science majors, but this is not to say that you may not be able to follow the same path that I follow today. And I would like to thank the previous speaker. I dreamt about it, and it happened. Thank you for encouraging the young people. What I would like to do today is to take you back in time to 1980s, and I'm sure most of you in this room were not born then, and I will gradually bring you up to date and to where we are today. So I initiated my education as an undergraduate in the university called Bosphorus University in English and Boğaziçi University in Turkish, which is located in Istanbul, Turkey. This is one of the Ivy League universities in Turkey where a lot of students have dreams about studying in, and I was kind of lucky enough to have a place there to initiate my studies. So I started my studies there in 1980s, towards the middle of the 80s, as an undergraduate student, and then I went on doing my master's degree in molecular biology and genetics, and then pursued along the same line to finish up my Ph.D. degree in 1995, the month of July. So during these times, what I had the chance of being occupied with was that I had the opportunity to work with different samples coming from all over Turkey to study the beta-globin gene mutations, in other words, beta-thalassemia mutations in the Turkish population. So I was one of the lucky ones to be part of the very first reference lab in Turkey, which was initiated under the guidance of Boğaziçi University. And we were in the group where we actually took care of the beta-thalassemia patients, sickle cell anemia patients, and there were other groups that actually worked on Duchenne muscular dystrophy, hemophilia, and whatnot. During my education as a graduate student, I had the opportunity to work on thousands of Turkish patient samples, and what you see on the slide here is showing the map of Turkey where we have identified unique mutations that were to do with the Turkish population. The next step in my journey was, after I graduated from my PhD studies, I started applying for fellowships in terms of pursuing further education. So as you notice, I did my undergrad, my master's, my PhD, and I didn't stop there. And today I haven't stopped either. You should know, those of you, I saw some people from my classes that I keep talking about my experiments, and although I am a lecturer today, I'm still a student, because there is no end of learning. You keep learning every day, on an everyday basis. As the technology progresses, we tend to learn as much as possible on an everyday basis. So what I did was, I applied to many universities, many research institutes after I graduated in 1995 from Boğaziçi University, and I got acceptance from several different institutions as well as universities, but I actually chose the one, well, I kind of jumped the gun now, but I just wanted to share with you one of the publications, in fact, two of the publications two of the many publications, I should say, that we actually studied and then performed experiments on, and we published them in some good journals and some as book chapters. So the next step, as I was saying, was to pursue with my postdoctoral studies after finishing the PhD graduation. So what I did was, I kind of got accepted into several different institutions, but I accepted the fellowship or Fogarty Fellowship from the National Institutes of Health of the United States. This is a very prestigious fellowship, and I was in fact the first Turkish Cypriot to be given such a fellowship, so I am actually honored by this. What you see in this slide here is the very famous building number 10 on the campus of National Institutes of Health. So I was actually located in this building, in the back one, on the 9th floor where my laboratory was. I spent my 6 years of time, and then I achieved a lot of success. I was very lucky to be there at the right time, right place, and I had the opportunity to work with very famous people in the field of my own interest. So these opened up a lot of doors for me in terms of having connections for future work, and you will hear me talking about some of these opportunities later, but just to continue on this line, I continued along the similar line of my PhD thesis. As I mentioned, I was working on globin genes. I was working on beta-thalassemia patients and finding out the prevalent mutations in the Turkish population. So that was one of the main reasons that my mentor in NIH accepted me to work with him. So what you see here is I went on working on further possibilities for finding out therapeutic means to cure beta-thalassemia. So this was one of the main reasons why I wanted to join my mentor's team. So for that, I started working with, so this was one of the projects that I started working with. I started working with mouse embryonic stem cells. You must have heard about the stem cells in general. I was not able to work with the human stem cells at the time because it was prohibited, because we were funded by the federal government and they were not supporting for us to be working with the human stem cells. It would have been more matching towards the cause of what we were trying to do, but mouse being a good model for studying human diseases, we started working with mouse embryonic stem cells. So we achieved many success along this line, and we published, one of the publications were to do with the genetic disorders of hemoglobin, and what you see here are the worldwide prevalence of beta-thalassemia mutations, in some parts sickle cell anemia, and we see, what you observe here is there were some methods that have been used towards identifying these mutations, whether it be radioactive methods or non-radioactive methods. So we actually compiled this whole work in a review article with my mentor at the NIH. The next step for me, after spending several years at the NIH, was to move on, because there's a certain time frame that you can spend as a fellow at the NIH. Depending on the visa that you are on, you are either required to go back your own countries and come back to United States if you want to continue, or you apply for a green card, for example, and then if you get accepted, you can pursue as a working person in the United States. So I actually obtained the green card. At the same time, I applied for an adjunct professorship at the University of Maryland, University College. So I started my first academic experience as a teacher when I was still working at the NIH, and then I moved on to working, I got an acceptance working as a legal worker per se. I was no longer a fellow, so I moved on within the same town of Bethesda, Maryland. I moved to University Services, University of Health Sciences, Uniformed Services, University of Health Sciences. This was a military university, but civilians were also accepted to work there. So I started working in the molecular biology department, and this time I kind of shifted my gear towards working in another discipline of science, which was cholesterol biosynthesis. What we did here was we were actually working on how cholesterol is produced when it is needed and how it is not produced when it is not needed. So these pictures, these colorful pictures that you're looking at just shows that. I won't go into detail, but just to give you the cream of the cake, this is an experiment done on signaling of cholesterol-related proteins. The next step, as I was telling you about making a lot of networking while I was a postdoc at the NIH, I meet a lot of people, I made friends, and the time came for me to again change my gears into a different discipline. So along those lines, I was actually offered another position as an adjunct professor at the Arizona State University. So I started also giving lectures and then kind of guiding, mentoring some graduate students. While having been offered a job in a non-profit organization in Arizona, Phoenix, I started working at an institute called Translational Genomics Research Institute. In short, we call it TGen. This is one of the known entities in the United States, famous for dealing with high technologies to try to find cure for cancers in general, also finding cures for neurodegenerative diseases and some rare disorders. So again, I was lucky to be part of a group to start working on now cancer-related topics. So originally, if you remember, I started telling you that my first step of being a student initiated with working with beta-thalassemia, sickle cell anemia. Then I went on doing some work on cholesterol biosynthesis, and then now I'm shifting my gears towards cancer. So I was kind of, I had the opportunity, and I was lucky enough to have a broad spectrum in science now, where I could kind of look from different angles to try to be able to find therapeutic means towards different diseases in life. So what you see here is, on the left-hand side, you see a biosynthesis of the cholesterol itself, where I was actually accustomed to a year before I got accepted working at TGen. So I applied to go to a workshop, and to my surprise in that workshop, it was a cancer workshop, but somebody started talking about a gene which was related to cholesterol biosynthesis. And guess what? That was the gene that I was working with. And I was kind of very surprised, astonished that, okay, this is my lead to start stepping into the field of studying cancer genetics. So I took that gene and I did several experiments to show that the gene of my interest was being differentially expressed in different cell lines that kind of represented cancer, that we could study the cancer in humans, okay? So that was very interesting for me. The next step was to, obviously, the reason why I was hired to work at TGen was to be part of the team that was going to work one of the very interesting and one of the novel technologies at the time, what we call gene silencing, RNAi mechanism. So what this methodology involved was, in science, in genetics, what we do in order to understand the function of genes is that to create mutations in those genes or to change their function. If the function of the gene seems to be kind of important, significant for the viability of the cell that you're working on, then you would see the cell dying, not proliferating, which would be a good way of representing how to attack tumors, how to attack cancer cells to get rid of them. So this method that I'm talking about right now is a method to silence genes one at a time to actually help us understand how can we resolve this deleterious means of cancer to get rid of the cancer in humans. So originally, obviously, before jumping to humans, because of ethical issues and because of norms that we need to follow in science, we need to start working on cell lines. Then if we're successful, we start working on animal models. Then if we're successful, we pass on to working with different groups of humans with their consent forms being signed. And then eventually, if everything goes well, then whatever gene or whatever drug you're working on, you can actually put it to the market for the utilization of specific diseases that one wants to work with. This particular example represents a cell line which actually is to do with breast cancer. So one of our experiments was to do with testing these cell lines, which actually represents the breast cancer in humans, to see whether the technology that we're using can help us silence certain genes and towards therapeutic means of creating some sort of a drug. We were successful in identifying some genes that actually would be potentially used as factors to kill breast cancer cell lines. So we actually published along these lines. This is one of the book chapters that we have published. This is another book chapter along the same line. This is a paper manuscript that we have published. In fact, this is on not breast cancer, but colon cancer. And if you remember, I talked about cholesterol biosynthesis. That information being in the background have been brought to the front burner of the stove this time. So I started using drugs that are used to lower cholesterol synthesis, but indeed not for cholesterol this time, for cancer, colon cancer to be precise. So we have obtained good, valuable results, and we have published it in PLOS ONE magazine. This is another chapter that we went on working on pharmacogenomic type of studies and published it in another book. So time passed. In fact, in 2011, I started thinking about coming home, home meaning North Cyprus. Then I have done all the things that I wanted to do. I was satisfied with my work. Then I thought, okay, it's time to give it back. When I say give it back, it is time to kind of mentor the young generations of our country and generations of other countries who joined to Eastern Mediterranean University. And I'm glad to be part of this university. This is our laboratory, which was established about three and a half years ago. It is up and running. We have more than 250 students now. We are going to have our first graduates in June, and I'm glad to be part of that team. And in fact, it has not been officially announced, but we are accredited by a German institution, AHPGS. So those of our students who are going to be graduated this year, their diplomas are going to be recognized worldwide. So this is a good news and new news for everybody. So I would like to thank you for your attention, and again, I am privileged to be here today. Thank you all for your attention.